Prokaryotic Cell Development: The Dimorphic Lifestyle of Caulobacter crescentus by David Sterling of Wayne. When we think of bacterial cell division, most people may think binary fission, and without directly saying it, we picture one cell dividing into two symmetrical daughter cells. This is not always the case. Although there are other forms of asexual production, we will focus on symmetry. One gram-negative bacterium, Caulobacter crescentus, has an asymmetric cell division that is quite analogous to how mammalian stem cells develop. When we observe Caulobacter, we see one cell type, the immobile or non-motile stalk cell, divide into a stalk cell and a motile swarmer cell. In all cases of asymmetric cell division, we see the pattern. A makes A plus B. The similarity between stem cells and dimorphic bacteria for our purposes ends here. Both have their own wonderful complexity. The team that had propelled research into the dimorphic life cycle of Caulobacter was a Canadian named Dr. Roger Yates Denier and his American mentee Dr. Jean Poindexter, both from UC Berkeley. Their landmark paper, published in 1964, called The Biological Properties and Classification of Caulobacter Group, as well as a follow-up paper in 1966 titled The Development of Cellular Stalks in Bacteria in the Journal of Cell Biology, was crucial in establishing Caulobacter crescentus as a model organism to study dimorphic replication. But before we begin, there are a few descriptions that might not be too clear to some. First, there are holdfast. They are exactly what they imply. Holdfast are adhesive structures located at the base of the flagellum and dividing in swarmer cells, and at the tip of the stalk in a stalked cell. Peptidoglycan are rigid layers of cell walls which comprise of a thin sheet of an N-acetylglucosamine, an N-acetylmuramaric acid, and a few amino acids. The stalk is an outgrowth and continuation of the inner membrane, peptidoglycan, outer membrane, and inner core of a membrane from which the cytoplasm is excluded. The MCP proteins monitor the concentration of various substances over time. These sensory proteins are called methyl-accepting chemotaxis proteins. In the Caulobacter life cycle, free swarmer cells alternate with cells that lack flagella and are attached to the surfaces by a stalk with a holdfast at its end. The role of a swarmer cell is dispersal, as swarmers cannot divide or replicate their DNA. The reverse is true for stalk cells. These are capable of reproducing. Think of a tree. If a tree reproduces and remains in the same field, seedlings have a difficult time trying to outcompete the older and more established generation. However, if a seedling grows and is moved to a field that's not populated by older trees, then that seedling can grow and survive. Additionally, if a wildfire destroys the older field of trees, the species still survives in a new location. It's easy to think of cell differentiation as a step-by-step process. However, in reality, many changes are happening throughout a cell's life at the same time. For Caulobacter crescentus, we can see those changes directly as a flagellated cell becomes a stalk cell. Since stalk cells are essentially mature flagellated cells, let's begin with how a swarmer cell becomes a mother cell. About a third into its life, in response to an unknown signal, the swarmer begins its process of shedding its pili and flagellum. The stalk is created at the same place where the flagella was once anchored. The first measurable change we can observe is the nucleoid changing from a tangled mass of DNA to a simple extended form. This usually happens when there are enough nutrients in the system. This extension, of course, is in preparation for the binary fission that will eventually take place. At the same time, a lot of changes are happening, like the degradation of CTRA proteins, chemotactic proteins, and pili. But let's return to the flagella and stalk. CTRA, by the way, has many functions, one of which is preventing the ability for DNA to replicate, so we can understand why it has to go. The cell cycle is controlled by three regulatory proteins: CTRA, GCRA, and DNAA. These are all transcriptional regulators, but DNAA has the extra task of initiating DNA replication. CTRA, once phosphorylated, activates transcription for flagellum synthesis from about 50 genes. Known as the global response regulator, CTRA activates other proteins such as sigma 54. Sigma 54, in turn, begins activating transcription of the basal body and hook proteins with help of a phosphorylated transcription factor called FLBD. DNAA binds to the origin of replication and initiates DNA replication by opening the double helix. Because CTRA also represses DNA replication, DNAA levels rise only when CTRA is degraded by a specific protease. Therefore, it's easy to see why there are high levels of CTRA in the swarmer cells and high levels of DNAA in stalk cells. The GCRA regulator promotes the elongation phase of chromosome replication, cell division, and the growth of the immobile daughter cell. Eventually, GCRA levels fall and high levels of GCRA reappear in the daughter cell, destined to swim away. The flagella is composed of a filament, hook, and rod substructures. In order to become a stalked mother cell, the flagella has to be jettisoned. This cut occurs somewhere in the rod. How this cut is made and the regulators for the injection process is still unknown. One suggestion from researchers from UCLA proposed that the breakdown of an anchor protein called FLIF is responsible for flagella loss. The cell begins to redirect cell surface proteins to synthesize the cell wall where the flagella once stood in a perpendicular direction, but only at the proper time and also relying on the extracellular phosphate concentration. You might be wondering, how does the cell know where to grow the flagella? A suggestion from researchers is that the localization of MCP on the polar end of the dividing cell attracts FLBD, and that a protease called FLIF could have an important impact where and how flagellum is made. A major goal of future research will be to identify the signal transduction pathways of Caulobacter that are due to environmental or internal cues. Thank you for listening, and hopefully you learned something about the double life of Caulobacter crescentus, this awesome gram-negative bacterium.